which will describe the active immunity. So the active immunity is involved that your bodies produce your own antibody. So it means the antibody is just from your body, it's not from the external source. So actually the answer is A. Okay, so it depends on phagocyte also. So they will produce the memory cell. Okay, means that after they produce the antibody, so the antibody they will bind to the antigen, right? Forming the antigens antibody complex. Forming this antigen antibody complex. So after that, the phagocyte will come. Okay, the phagocyte will come. So they will engulf the whole antigens antibody complex, and then they will digest it. So this is we call as the phagocytosis. So it's a part of the process in the active immunity. Okay, it's a part of the process in active immunity. Then how about how memory cell is being produced? So it's something like this. You can imagine that. Means like this is the antibody, right? Antibody is a Y shape like this. Okay, just a Y shape. So after, when they are binding to the antigen on a certain pathogen, for example, maybe they are binding to a bacteria. Okay, I draw a bacteria here. The bacteria, they have the cell surface protein, right? Or we call as the antigens. Okay, like this. This is their antigens. This is a bacteria. And this is antigens on the bacteria. So antigen is part of the pathogen itself. Okay, antigen is part of the pathogen, which is the antibody binding site on the pathogens. So they know antigens. So when the antibody they bind to the antigens, they form the antigen antibody complex like this, right? Because antigen antibody complex. So the reason they need to form this complex is because that antibody, they have the ability to agglutinate. We call it agglutinates or clump. They can form a clump together, okay? Together with another pathogen. They just clump them together. So it's easier for the phagocyte to do the phagocytosis later. Okay, so by this process, they will also stimulate the production of memory cell. So how they produce the memory cell, okay? So initially they will they will have this lymphocyte, okay? So this is a lymphocyte. Lymphocyte is like this. Okay, lymphocyte has a very round nucleus, round and prominent nucleus. So there will be two functions of a lymphocyte. First, their function is produce what? Lymphocyte, they produce the antibody. Remember, lymphocyte, the first function is they will produce the antibody. AB means antibody. Secondly, they will produce the memory cell. So actually this lymphocyte, when they are under attacks by the pathogens, lymphocyte, they can divide so fast. Okay, they can divide very fast. Means that from one lymphocyte, they can do the divisions, okay, divisions to produce a lot of lymphocyte. A lot of lymphocyte, maybe from 1 become 10, 20 like that. So then those lymphocytes can differentiate, become the memory cell, or they will produce a lot of antibody like this. So the antibody, the memory cell. So the memory cell, they will give the long-term immunity. Okay, they will give a long-term immunity. So if there's without the presence of memory cell, there will be no long-term immunity. Okay. So I think other is correct already. Yeah, I think other is correct. I think it's simple chapter for you, okay? Because both of you get almost correct answer. Yeah, that's all finished. Okay, so today we go through about how do we answer for the chapter 10 paper 4, okay? So now you can open this note. Oh, sorry. You can open this note. You open this note. You open this chapter 10 paper 4 answer. I do share you in the WeChat. I think this, yeah, chapter 10 paper 4. Okay, you open this PDF and then I'll explain to you. Just want to show you that what kind of questions usually come out in the paper 4, and how do you able to answer the paper 4? Okay, once you're ready, let me know. Once you already open this file, let me know.
Ready.
Okay, sure. So for this chapter, tense immunity, you can see, see the genes for the antibody are only active in the lymphocyte. Okay, so this means what? This means that the antibody, because antibody is a kind of proteins. Okay, I hope now you already know that antibody actually is a kind of protein, just same to your enzyme. Okay, same to your hormones. They are example of protein products, right? Okay, they are example of protein product, protein O products. So in order for your body cell to produce the antibody, they also need to undergo the transcriptions and the translation that we learned before, which is inside the nucleus. They contain the DNA. The DNA contain what? The DNA contain the genetic material. Okay, so like this. Okay, this is a cell. This is a cell, and then this is a nucleus, right? This what I'm drawing like this. Sorry. Okay, I think I draw a wrong image for you. Okay, just imagine that this is a nucleus, right? This is a nucleus. So inside the nucleus, there's a lot of DNA. Okay, so the DNA will be in the chromatin form, because it's that long thread-like DNA. Okay, chromatin means like thread-like DNA structure. Just looking like long thread and quite messy. Thread-like DNA structure. So when they it means that when these long threads, they can further undergo the condensations. Condensation means that they will combine. Okay, so after they combine, they will become chromosome like this. This is a chromosome, okay? So they ask you to define what's gene. Okay, gene, this you will learn more in chapter 70, which is the inheritance. But you just need to know that, okay, genes is just a portion of the DNA that encode for proteins. You can imagine gene is like in this way, okay? For example, gene something like this. This is the DNA, right? See? So DNA is very long and complex structures without any ending. This is DNA. So what's different DNA and the genes? The difference for the DNA and the gene is this whole thing is DNA. How many strands in the DNA? Double strand, right? So DNA we always call as double-stranded or double helix. Double-stranded or double helix DNA, okay? So this whole thing is DNA, no ending one. So now you want to show where's the location of gene. So gene, maybe they can be like here. This portion is a gene. Okay, this portion is a gene. Maybe you call it gene A. And then these portions, they will be the gene. Maybe you call it gene B. This portion, maybe we can call as a gene C. So this show what? This show that gene, they refer to the portion or a short length of the DNA. Okay, a short length of DNA 就是在这个整个 DNA 一整端的一小段而已。OK， 一小节。OK， 一小节。So this gene, okay, different genes which occupy on different locations. See, different gene which occupy on the different locations of the DNA. Let's see, it's upper parts. This is the middle part. This is the lowest part, right? So different regions, but on the same strand of DNA, right? Different region, but on the same strand of DNA. So those genes actually they are encoded for different proteins. For example, this gene A, they are encoded for protein A. Okay, for example, protein A, what kind of protein A they encode? Maybe they are encoded for the, for example, like hair color are like this. Okay, so for example, maybe this gene B, they are encoded for the skin. Protein B, which is the skin color. They give you, they they will give you the color of your skin cell or something like that. So different function, okay? So maybe this gene is related to the length, for example, the height. So this protein C will be related to height. So you can see different genes with different function of proteins, but they are located on the same strand of DNA. So this we call a gene. Okay, so back to the questions. So what is meant by gene? Gene is the length of DNA that encodes for proteins. Sorry, okay. Gene is the length of DNA that encodes for proteins. So you can see this encodes, right? Encodes. What is meant by encode? Encode is means like it's because that you still remember that last time we learned about transition and translation process, right? In chapter 1. So actually they are trying to they are trying to do the transition from the DNA, okay, from the DNA, the nucleotide base, from the nucleotide base sequence, nucleotide base sequence in the DNA. And then now become what? Then become the amino acid sequence in the change, in the polypeptide change. So after that, they will become the different protein structures. And functions also. Okay, as the function of protein is determined by their own structure. So this is meant by this okay, length of DNA, they are encodes for proteins. Then for this one, okay, lymphocyte produce the antibody, so they ask you to outline, okay, they ask you to outline the role of antibody in the defense of the body against the pathogens. So how the antibody do on their roles. So antibody, they usually able to bind to the antigens on the surface pathogens. So they will form the antigens antibody complex. After they form this complex, okay, maybe we should understand that how they able to form this complex is because that their structure must be complementary to each other, just like how the substrate able to bind to the active site on the enzyme, right? So it means that the shape need to be complementary. They can fix to each other. So that means that antibody and the antigen, they have a very, very specific shape or structure. So antibody, they have a complementary shape. So you can see again, this keyword is being used again. Complementary shapes to the shape of antigens. Okay, so this will cause the antibody able to bind to the antigens on the pathogens. Maybe you can add something like this. Okay, antibody able to bind to antigen on the pathogen. No matter it's what kind of pathogen, either it's viral or DNA. So antibody, what they do, actually they will agglutinate, clump the pathogen together. So actually they can help to destroy the pathogens also. Okay, they can help to destroy the pathogen. Some antibody, they able to neutralize the toxins being produced by the pathogens. Okay, this you will learn more. You will learn more about the function of the antibody in a more detailed way. Okay, you'll be learning this in the advanced level biology, like A level or IB biology. You learn more because antibody have that. They have their own function also. So they can destroy the pathogens. Okay, so usually what they do is they mark the pathogens, just like you highlight the important point, right? So you highlight so you can allow other people to see it. So they highlight, okay, or they mark the pathogen, so they allow the phagocyte, when they come in, they just easily see and they can just engulf it very easily. They can destroy it by the phagocytosis. So this is the answer. 4 marks, okay, 4 marks. You should write about the antibody, they bind to the antigens, right, on the surface of pathogens, forming the antigen antibody complex. 2 marks already. And then you should say that they have a very specific shape, so they are only complementary and destructions by the phagocytosis later. Okay, so remember this one. They mark the pathogens, or you can say that antibody, they will agglutinate the pathogens together, so to mark for the destructions by the phagocytosis. Also can up to you. Just you need to put the keywords include in your answer. Okay, so this one, this is the lymphocyte, they produce the antibody. Okay, so I had labeled out for you. Okay, just can identify the structure. Okay, this is nucleus, right? Nucleus and the ribosome, and then cytoplasm, and then this E is the mitochondria, because they has a highly folded inner membranes. Okay, you can see inside the inner membrane is highly folded, and then the small vesicles. So they're asking about what? Oh, okay, this is asking about the structure. Okay, I think you already know how to do it. Okay, but see, provide energy for making antibody. So you can see that when the cell need to produce antibody, right, they require energies. So where they can get energy? The cell, they will get the energy from the cellular alveolar respirations. They will get this energy. You can see making antibody. Making antibody is a process which is require input of energy, because making antibody is a process which is considered under the, we call it as anabolism, right? Anabolism means that they will produce something, synthesize on new thing, a new cell. Anabolism, which is a part of your metabolism. Okay, anabolism is a part of the metabolism, right? Metabolism is a refer to the all chemical reactions that occurs in your body cell. All chemical reactions include your anabolism, which is you're trying to building something, or include catabolism, which is you're trying to breaking something. Okay, so yeah. So site of production antibody. So you can see antibody is a protein, right? So where is the site of production of protein? So the answer would be the ribosome. Or you can write the endoplasmic reticulum. But you writing the ribosome is better for you, because ribosome is the organelle, which is they will produce the proteins. Ribosome, they will produce the proteins, which include the antibody, they include the enzyme, hormones, and other kind of proteins. Okay, so after they already finished producing the protein, right, from the Golgi apparatus, so the way how they produce the antibody, just like similar the way how they produce the proteins, right? So initially they will be like the DNA inside, right? See the DNA inside. So DNA, they will do what? DNA, they will do the transition. So they will produce what? They will produce the messenger RNA. So remember, messenger RNA. So the messenger RNA, they will go out from the nucleus to the cytoplasm, and then they will get contact with the ribosome. So the ribosome, they will be reached along the messenger RNA, so they start to produce proteins. So how they do? They will join the MSC together to form the polypeptide chain. Yeah. Okay, that's all. I think that's all. So after they start to produce the polypeptide chains, okay, after they will folded, and then they will go into the Golgi apparatus, Golgi apparatus, which is here. Okay, tell me what is function of Golgi apparatus? Golgi apparatus function is do what? Their function is they will try to transport packaging processings. Okay, for example, because when the proteins, they just come out from the ribosome, it's just protein only. It's not functional protein, right? It's just proteins. You know the difference between the raw protein and the functional proteins? Yes, ribosome, they do produce the proteins, but usually the proteins is just like a raw material to make another useful protein products. Correct. So after that, proteins, they will need to transport by RER. Where is my? Okay, they want to transfer by the RER to Golgi apparatus. Help you to revise for the chapter 2. Okay, so after that, the RER, oh no, the Golgi apparatus, what they do is they will help to modify, packaging, processing. Those proteins become the functional protein product, which is, for example, they become the antibody now, which is the functional proteins. Means they can really carry out the functions. So the antibody, after they've been produced from the Golgi apparatus, they will be released in the form of vesicle. Okay, see vesicle. So the vesicle inside they connect antibody. So vesicle they will fuse to the cell membranes and then they release antibody out. They just release and go out. So the antibody can go to find their pathogens. Okay. So you can see this vesicle is being formed. So actually vesicle there contains two types of vesicle. I hope you still remember. So this vesicle, you can see, they are trying to transport the products out. It's called as a secretory vesicle. Secretory vesicle. Because they help in the secretions. Secretion. Okay, for this one. Say the name of one type of cell other than lymphocyte that is involved in the defense of the body against a pathogen and describe its role. So the phagocyte can eat the pathogens. The phagocyte, they can eat the pathogens. Okay? Phagocyte, they can eat the pathogens. So now what they do is they will engulf the pathogens, bacteria, and virus. So how they do is, yeah. So this is called as phagocytosis. Because the question asks you about what? The question asks you about state the name of one type of cell other than lymphocyte. Because you have two white blood cells learned in your IGC biology, right? Just remember, there's only two white blood cells. One is phagocyte, one is lymphocyte. Lymphocyte usually their function is to what? Antibody. They'll produce antibody also, and then they will produce the memory cell. Usually this function. Phagocyte is just they just eating. They do the phagocytosis. So they eat those kind of cells, see, pathogens, bacteria, virus, yeah. So they help with the immunity defense. So you can see for this one, you need to identify what is this cell. Okay, so what is this cell? S. S is the lymphocyte because they have a very big and prominent nucleus. Then how about T? T is referred to the bacteria. Okay, T is referred to the antigens. Antigens on the bacterial surface. It's referred to the antigen for the antigen on the bacterial surface. Yeah. So you can see that this is a T, right? T is the antigens. And then this U, okay. So you can see this process is doing what? This process actually we call as, you see from this S is a lymphocyte, right? You can see lymphocyte, they divide, become 2, then become 4. So this is how the lymphocyte able to produce a lot of lymphocyte in a very short period. Okay, so actually this is term, okay? So you can see after they produce a lot of lymphocyte, they are increasing the number of lymphocyte, right? So what they do, each lymphocyte they produce more antibody. So when you increasing the number for the lymphocyte, actually you are increasing the number of the, actually you are increasing the amount of the antibody productions. More antibody they will produce. Okay, so this will enhance the immune response because we want to produce a large number of antibody just in a very, very short time only to overcome the pathogens, right? So they tell you R is prokaryote and S is the lymphocyte. So state the name of two cellular structures they could found in both prokaryotes and the white blood cell. So you can see that this prokaryote, okay, see this prokaryote. Prokaryote both contain the ribosome and also both contain the cell membrane and cytoplasm. So actually this is, they ask you to compare between the difference of the prokaryote cell and the eukaryote cell. This is a very common question asking in the exam. Okay, see, because white blood cell is a eukaryote cell, okay, eukaryote cell. Same to red blood cell. Red blood cell also a eukaryote cell. Even though red blood cell they don't have the, they don't really have the nucleus. But initially they have the nucleus, so just that during the maturation of the red blood cell, they remove away their nucleus because they want to carry more oxygen, right? But they are still belong to the eukaryote cell. Okay, so the similarity for the prokaryote cell and the eukaryote cell is what? Is both contain the DNA also. Even though in the eukaryote cell, what kind of DNA is a eukaryote cell? It's a linear DNA. Eukaryote cell is DNA, linear DNA. Prokaryote cell is a circular DNA, but both also contain DNA. You can write this become your answer. Okay, they have a ribosome also, yeah, like this. Okay, then number 2, they ask you to do what? They ask you to describe the cell structure of the white blood cell different from the cellular structure of prokaryote. Yeah, just similar thing. Yeah, okay. What's different? The difference is the white blood cell, they contain nucleus. So the prokaryote cell, they don't have nucleus, just like this. Okay, so the white blood cell, they do have the membranous organelle, but the prokaryote cell, they don't have the membranous organelle, just like this. And also prokaryote cell, they have the plasmid. White blood cell, no plasmid. Just answer in this way. Very simple. And then for this one, cell R is a pathogen that has structure T on its surface. So this structure are recognized by the cell S. Then cell S is a lymphocyte and it produces a structure V. So you should recognize about this. See, those are the labels that you should recognize. Okay, see, cell R is a pathogen, right? So R, they might be the either it's bacteria or virus. Yeah. Then structure T. Structure T is antigens. Okay, they're recognized by the S. S is the S is the lymphocyte. They tell you S is the lymphocyte. So lymphocyte produce the what? They produce the antibody. So lymphocyte, they produce antibody. So cell R reproduced by binary frictions and the cell S divide by the process U. So it's cell division. So you can see when the lymphocyte produces a lot of lymphocytes, so this is known as cell division. Actually, when you go to the advanced level of biology, so this is not cell division. This is actually, they call as what? They call as a clonal expansion. This is just for your information only, okay? Clonal expansion. They expand to increase their number. But you just need to know the cell division only for the sake of your exam. And then for this one, okay? This one, a lot of students, when they look at this picture, they can't recognize what kind of the cell is it. So actually, it's belong to phagocyte. Okay, it's phagocyte. So how I know it's phagocyte? It's because that you can see this cell, they are trying to engulf on something. This is cell W, right? This is pathogen. So they must be the phagocyte because only phagocyte, they do the phagocytosis. Other cells don't, right? So describe how happens. Okay, what happens? Phagocytes, they engulf. Okay, you cannot say it. You cannot say phagocyte eat pathogen. It's not professional for you. You should write phagocyte engulf. Engulf refer to cell eating. Refer to cell eating in the cell eating in the way. Sorry, yeah. Refer to cell eating. This one, cell eating. Yeah. So you should write engulf. Not eating. Eating is refer to human and animals, but cell is engulf. Means that it swallows the pathogens. So they will form the phagocytic vacuole. See, phagocytic vacuole like this. Phagocytic vacuole is just something like the food vacuole. Yeah. So inside the phagocyte, they contain the enzyme. They contain we call it as a lysozyme. Okay, it's a kind of hydrolytic enzyme. You can say it's a hydrolytic enzyme. Just similar to our digestive tract. In our digestive tract, we also have the enzyme, right? We have the protease lipase or something like that. So they help to break down the pathogen. So the highlighted is a keyword that you should write in your answer. For this one, meningitis. Okay, meningitis in Hua is called as 脑膜炎. Usually it's caused by bacteria. The bacterial infection infect we call it as your I forget the structure, but meningitis 脑膜炎, okay, is infected to the membranes, okay, the membrane cover on your brains. Yeah. So it's a kind of contagious disease, okay, or we call it transmissible disease. So what is the definition? Is able to transmit, okay, from the infected to the non-infected people. So this is the keyword. What's meant by transmissible? Infected to non-infected. So they made the non-infected become infected already. So how they cause this is usually caused by the agents, right? The pathogens, such as the virus, bacteria, parasite. Yeah. And then this one. How vaccination provide the active immunity is because that vaccine, they contain very weakened pathogens, right? You can say the pathogens, or you can say that attenuated pathogens. also. So actually, inside the vaccine, they do have the pathogens. They do have the virus, right? So last time you still remember, we do the coronavirus injection. Okay, you do the coronavirus injections. So the coronavirus injection, they also, I mean, the vaccine. Okay, let me close my WhatsApp. So noisy. I'm so sorry. The coronavirus vaccine inside, they have the coronavirus, but the coronavirus just maybe they just took the fragments of the coronavirus. They did not put the whole virus, but just fragments of the coronavirus. So because they want to allow the vaccines, okay, which contain the part of the pathogens, even though the pathogen is weakened already. So they must ensure that the pathogen is weakened, so they did not really cause disease to the healthy people. Okay, so they must be weakened first. Because the purpose is they just want to stimulate our body to produce the immune response. So this is the main this is the main purpose. So last time we do the coronavirus vaccine injection is because that we haven't we haven't infected with the coronavirus okay, again. So that's why we need to protect us by the vaccination first. We need to protect us by the vaccination first. So if there's actual if in the actual case we really being infected by the coronavirus, at least we already been vaccinated. So after we've been vaccinated, right? So our body cell actually they will have the memory cell, right? Memory cell. So if the same coronavirus infected us again, so actually the memory cell they will recognize the virus, so they can produce a lot of immunity. They will produce a lot of antibody in a very short time, just like you see just now, right? From one lymphocyte become 2, then become 4. Yeah. So lymphocyte make the antibody, so they can produce a lot of memory cell. So this will create a long-term immunity. So please remember to write memory cell and the long-term immunity. No memory cell means no long-term immunity. Means your immunity becomes short-term immunity, which is not good. Okay, you should always keep the we call as immunological response or memories in your body to protect yourself. Okay, this one. If the meningitis disappear from the country, explain why the vaccine should continue to be used in the country. Okay, if there's no more disease, for example, you see, is there any coronavirus now happen? Very less, right? Now the incident become lesser compared to the few years, compared to few years ago. But actually, we still encourage to do the vaccination. Even though there's no more disease happen, we still encourage. Why? Because bacteria, they may still present in the populations. Some people, they have no symptom, but they are belong to carrier. They are carrier, but maybe they don't know they are the carrier. So if too few people are being vaccinated, means that if too less people are being vaccinated, bacteria is more likely to transmit, okay, from the infected to the uninfected. So if you have more people vaccinated against this disease, so it will definitely help to reduce the bacterial transmission from people to people. So this is called as they are creating the herd immunity, right? Herd immunity. So do you know that last time when when we being impacted by the coronavirus, so after we do we call it as mass vaccination, right? Means that a lot of people they being vaccinated against the coronavirus. So after that, you can realize that the case of the coronavirus they decline very rapidly, because we already created herd immunity. How can we create a herd immunity? It's due to the vaccination. Just get more people being vaccinated. You can create the herd immunity. Protect yourself, protect your family also. So, and also, you actually protect the unvaccinated people. It's because that vaccinations have this kind of property. Okay, if you're being vaccinated, you will still get coronavirus also. Okay, you still get COVID. But even though you get COVID, your symptoms will be less severe compared to the unvaccinated people. Okay, because for the unvaccinated people, first time they're being infected by the COVID, right? Their body really, their immune response really unprepared. They did not face this kind of this kind of the attack before, right? So that's why they need time. They need time to generate the antibody. So during this period, the people will be very, very weak because their antibody is still very low. Okay, and then they are still weak. But if the vaccinated people, due to they are already being vaccinated before, inject with with the antigen before, right? With the antigen from the coronavirus before. So that's why when they do the, when they are being infected against, so their memory cell, okay, will quickly recognize, oh, this is actually from the coronavirus, so they can launch an immune response, a very, very immediate immune response to clear all the pathogens. Okay, this one, okay, suggests why the antibody must be injected rather than taking them by mouth. Why? Do you know why? Why we need to inject antibody, but we are not swallowing the antibody like we are swallowing our pills or medicines? It's because that antibody itself is a proteins. Okay, this one you can write. Antibody are made of proteins. So you can imagine that when you swallowing the antibody, what will happen? Just become the food, right? So the antibody, they will enter into the stomach. So what kind of enzyme content inside the stomach? Which is the pepsin, right? Yeah, it's a pepsin. So the pepsin will digest the antibody. So the antibody will disappear. So they will be digested by enzyme in the stomach. So this is not practical at all. That's why we need to inject the antibody. And this one, explain why the passive immunity does not give long-term protection. It's because that for the passive immunity, it's not your body produce the, required as the antibody. Yeah, it's not your body produce the antibody, but you get the antibody from the external source, right? So this means that there's no active immune response and there's no memory cell being created. So antibody are just broken down in the body. Okay, even though you do injections, you inject the external source of antibody to your body, right? So the antibody, they have the half-life also. You know half-life. Half-life. Half-life means that they only exist for the certain period only. For example, the antibody, they can exist for about, their half-life is 5 days, right? So means that the concentration of antibody, they will reduce from 5 days, become half after, they will become half after 5 days. Yeah. Until they become totally zero at all in your blood level. So due to antibody, they can be broken down in the body. Your antibody itself is protein, right? So they can be digested by other cells. Yeah, so you really cannot depend on this passive immunity. You still require your body to produce your own active immunity, to produce your own antibody. Okay, for this one, outline the body defense mechanism is mechanical and chemical barrier. Okay, you can write. Mechanical barrier is referred to the hair in the nose. Chemical barrier is referred to the enzymes, hydrochloric acids, those kind of things you learned before. So also that apart from these mechanical chemical barriers, and also you can talk about lymphocyte. You can talk about the lymphocyte roles, right? The lymphocyte, what they do is they produce antibody also, and also they produce a memory cell. Please include the memory cell, for the long-term immunity. Yeah. This one, okay. This one, you need to recognize which one is phagocyte, which one is lymphocyte. So phagocyte, you can see that their nucleus is always just like, we call it as lobe nucleus, L-O-O-B. This one, L-O-B-B-E-D. The nucleus just look like lobe, one lobe, two lobes, third lobe, like this. So usually it's belong to the phagocyte. Only phagocyte has this kind of characteristic. Then how about this one, lymphocyte? Lymphocyte, the antibody is like circle, round and prominent. Yeah. Okay, so you should recognize this Q. Q is a pathogens, right? It's pathogens. Pathogens, they have antigens on the surface. So S and R both are the white blood cell, just that one is phagocyte, one is lymphocyte. So you should say that this cell S, right? S, which is a lymphocyte, they will try to make the antibodies. So the antibody, they have what? They have a very special shape. They have a very special shapes, okay, which is complementary to the antigens on the pathogen. See? So forming the antigen, antibody complex. So produce memory cell. So it's just very simple, okay? That's actually very simple. And then for this one, this is a graph, okay? So actually, this kind of questions, usually they'll come up at least one. I think maximum is also one question only. So you need to learn how to describe the trend of the graph, especially in this way. Look very complicated, right? But you can see this one. Okay, in 1946, the government of the country concluded that the vaccine was successful. So you must show that, see, discuss the evidence, you agree with this statement or you against these conclusions. Okay, so you should write these 2 points. What made you agree with this conclusion and what made you against to this conclusion? So you can see that to support the conclusions from 1942 after introduced the vaccinations, so you can see start from 1942 after they introduced vaccination, what happened is you can see there's a rapid decline, right? Rapid decline for this line from here to here. You can see my cursor, right? I hope you can see my cursor. See this drop to here, such a massive decrement. Okay, so general decrease in the case, so the case do not return to pre-vaccine level because before they do the vaccination, right, the case number is really very high, pick up. So after they do the vaccinations, you can see the case number significantly reduced already. So to against the conclusions means that you not agree, right? You not agree. So what made you not agree is because that even though you have a massive decline in the case, about from how many years? I can't see how many years. 1935 like that, maybe 1937 to 1940s. But after 1940 above, there will be slight increase in case. See, increasing in case number. So you can say that the number of cases increased during 2 years after the vaccine was introduced until the government makes conclusions. Yeah, see, increased for 2 years already. See, 1, 2, 2 years. So actually, they say that you not support this conclusion is because that the case increased, even though they just increased just for a little time only, 2 years only. And also, that means that there are too many small pigs in the case. Just like that. Just you need to think some logical reasons to support your answer. That's all. And then for this one, they showed about the concentration antibody. So at that actually you can measure your antibody concentration from your blood. So every time you go to blood checkup, right? What they check about your red blood cell, the concentration, which is the hemoglobin, and also they check about how many lymphocyte count in your blood. So this blood will give you this kind of information. They actually also can calculate, not calculate, but they can estimate how many concentration of antibodies in your blood levels. They can do this also. Okay, why? Because they need to see how the immune response toward the certain pathogens, like how the antibodies concentrations can effectively reduce the reduce the disease happens. So this they call about diphtheria. Do you know diphtheria? Diphtheria is a kind of bacterium. So this bacteria usually will attack our inner mouth. Okay. So when they are being infected, so they will be given injection of the antitoxin antibody. So this antitoxin antibody is referred to what? It's referred to the passive immunity, right? Because you inject the antibody, which is not being produced by your own body cell. You just get from outside, inject to your body. So this means that this kind of antibody, they also did not last very long. It will decline. Okay. So, but you still need to inject antibodies because what? Because you need to provide requires immediate protections. You need to provide a very fast protections. Okay. Yeah. So you can see that this trend. Okay, this trend. See, this is a graph, right? This is graph, and then you can see this one. See, the concentration of antibody in the blood. So this is the first time when you get the vaccines, and second time you can see the first time the antibody they rise up very slow, not fast. But after you do the second injection, the antibody rise up very fast, so fast. Okay, so this is called as a line for the active immunity. Because vaccination, okay, at the well, please remember that vaccination is a kind of the active immunity. It's an artificial form of active immunity. It's not passive immunity, because a lot of students, they thought vaccination is the passive immunity. I don't know why they think like this, but vaccination is definitely an active immunity. Just like it's artificial, because you did not get the disease yet. You just purposely inject part of the disease agent into your body, just to stimulate them to produce the antibody memory cell, just to prepare first. So you can see when they when they are being injected with the antitoxin antibody, so you can see it starts from the first day to last day, right? So the level decline very rapidly until become 0. So okay, they still asking about advantages. So advantage of this, you are giving the antitoxins injections. Their advantage is you're able to stimulate the artificial passive immunity. Okay, it's a passive immunity, but it's artificial, right? Okay, when we talk about natural passive immunity, natural passive immunity is like you get this immunity in a natural way, not artificial. Injections is artificial form. Okay, natural way is maybe you're drinking something like maybe they are drinking the mother milks, okay, the milks of mother from the mammary gland itself. So by this natural way, they get the natural passive immunity from the mother. So the passive immunity, their advantage is they can provide a very immediate protection. See, immediate and fast protections compared to the active immunity. Active immunity, if you want to build a very strong immunity from the active immunity, from the active immunity, you require longer time, right? You require longer time, right? So the protection is not immediate. It's just very slow. Slow, but it's long lasting. Okay, it's long lasting. So antitoxin antibody able to neutralize the toxin produced by bacteria. You can see this is a functional antibody. Antibody, they are not just agglutinated, okay? They are not just agglutinated and bind to the pathogens, right? But they also can neutralize the toxins produced by the pathogen itself. Because pathogen, usually bacteria, they can produce the toxins. So sometimes these toxins, okay, means that when your bacteria infected to your body, right? So bacteria, they will produce the toxins. So sometimes these toxins they will cause an inflammation of your body cell. Means that your body cell, or your skin appear reddish, right? Due to the inflammation. 发炎, inflammation means 发炎. So they can help to neutralize the toxins. So this will give time for the immune system to produce on antibody by the active immunities. Okay, so we already know that if you want to produce an active immunity, right, you don't have much time, okay, because you need to wait first. You need to wait. See, first injection, you need to get second injection also. So that's why last time coronavirus, we get a lot of booster, right? How many booster do we get? I get 4 booster. Okay, every year. Okay, actually they are encouraged every year you should get a booster. Okay, because you want to remind. Okay, immunological response, they have the period of memory. If you long time didn't inject your cell, okay, means that sometimes the memory cell, they will decline also. Sometimes. So you need to do the vaccine booster. So the purpose of this antitoxin antibody is because that they know that the active immunity, they require time to build up first. So, but they still need to get the faster protections, right? So they need to inject with the antitoxin antibody first. So explain how the injections of the vaccine result in the better protections against the diphtheria than the injection of antitoxin antibody. So why inject vaccine better compared to inject with antitoxin? It's because that when you inject the vaccine, right, you're actually training your immune response. Okay, and also you stimulate your body to produce their own antibodies, which is much more long lasting compared to your external source or antibody. Okay, so in the first exposed to the pathogens, they will produce memory cell to create a long-term immunity protections. So after second injections, there's a higher concentration of antibodies than the passive immunity. So if your body able to produce a large amount of antibodies, so actually you help to prevent infected with the disease again. So usually second injection, you can see second injection, they can induce a greater immune response. See, you see the graph, right? second injections, the antibody level rise so fast. That's why we need to get booster. And then how the antibody protects the bodies against the pathogen. So yeah, just similar like this. Okay, same answer like just now. And then for this one, the function of the lymphatic system other than defense against the disease. Okay, just for information, I think I told you before that our immune system actually is closely related to lymphatic system. Why? It's because that our immune system require the white blood cell, right? Production of white blood cell, yeah, those kind of things. So actually they are found in the lymph nodes, lymph nodes. Lymph node is belong to the lymphatic system. Okay, so lymphatic system actually one of their function is they help to defense against disease, just like immune system. Lymphatic system they can help to defense against this disease because that in the lymph node, lymph, they contains the lymphocyte. Actually, the lymphocyte, they are staying inside lymph node. They're doing what? They're doing the cell division. So just how the cell division that you see just now, right? From one lymphocyte produce 2, 4, produce a lot amount of the, produce a large amount of the antibody. So actually, this happened in the lymph nodes. 发生在我们这个淋巴里面 okay? In our lymph nodes. So other than that, what kind of other function is they can transport the fatty acid glycerol from the lacteal because they contain the lymph, right? Lymph. They will drain the tissue fluids, return fluid back to the blood plasma. So if they did not drain excess tissue fluid back to the blood plasma, what will happen? If your lymphatic system, they did not drain the excess tissue fluid back to your plasma, the tissue fluid will still accumulate within your, we call it as well. We call it the intercellular space. It's not. Intercellular space, okay, which is the space between the body cell. So this is called the edema, okay, 水肿, edema. So to prevent edema happens, you need to drain excess tissue fluid from your body cell, from the surrounding of body cell. Okay, to prevent accumulation of tissue fluid. Then this one, yeah, it's a bacteria. It's a cholera, okay? I think you heard about cholera before, right? Yes. Suggests what the cholera vaccine must contain to be effective. So what are found inside the vaccines? So the vaccines inside, they have a very dense and weakened form of pathogens. So these pathogens, they can stimulate the immunity to produce a lot of lymphocytes. So to destroy the bacteria. That explains why the people are not protected from the cholera immediately after receiving for the vaccination. It's because that you can see just like shown in this graph. Okay, this graph is actually very nice, this graph. So from this graph, you can understand that why in my first injection of vaccine, I still get COVID. And also I still have a very severe impact. I get a vaccine already, or why? It's because that you're just newly exposed to the disease, right? Yeah. So you'll be, feeling better after you get the booster, because after booster, your antibody level will be another level, higher level. So as we know that all active immunity, not passive but active. All active immunity, they just take a very long time to occur. It's not very long time, but they take time. So you require lymphocyte to release antibody, which is just specific to the antigens on the cholera surface. So they will produce memory cell and long-term immunity. Okay, so other is just like this. And also, yeah, with this one. Okay, you have two groups, right? Vaccinated and unvaccinated. Diarrhea and the high body temperature. So you see the information describe how effective the vaccines was at preventing the symptom of cholera. So most pathogens discovered in the non-vaccinated group, right? Yeah, you didn't get vaccines. That's why you shed a lot of pathogens. So some vaccinated, they did get the symptoms. So vaccine is not 100% effective. This is very important. Don't think that after you're being vaccinated, you will not get this virus at all, or this disease at all. You will still get it, just like your symptom may be not really severe. Means that you recover very fast because your body have a large amount of antibody can bind to those pathogens. Okay, so that's all for the immunity system. Yeah, for the immune system. Okay, for the immune system. So maybe we can continue about the new chapter. Okay, for the new chapter, which is chapter 11. Okay, please open your chapter 11 notes. We learn about the gas exchange in the humans. How we do the gas exchange. So this is the process that we did every seconds of our life. Okay, we're always breathing, right? Because we are humans, we are living organisms. We always need the oxygens. Okay, so are you ready for this now?
Yeah.
Okay, thank you. Okay, see okay. Previously, I'm not sure. I think we learned about the respiratory structure, right? So means that whenever you inhale, okay, you can see this photo. Whenever you inhale the air, right, the air will travel to your nasal cavity first. Where is it? The air will travel to nasal cavity first. Nasal cavity is 鼻腔. So, and then they'll enter to trachea second. Okay? Trachea is a cartilage. It's a cartilage. So trachea is usually rigid, but it's flexible. It's cartilage. And then you go into the bronchus. Okay, bronchus you have two sides, right side and the left side, two side bronchus. And then you have the bronchiole here. This is called bronchiole. And then from the bronchiole, you reach alveolus. So those I label with number after pathway for the respiratory tracts. Okay, so you can see bronchiole will be here, just on top of the alveolus only. Okay, and then now we talk about the alveolus, right? Alveolus is the ending part of the lungs. Okay, because alveolus they are very closely connected with the blood capillary. Okay, alveolus, you can see their structure, right? This is the alveolus. This is the blood capillary. They just flow very close to each other, right? Because they want to easier for the gaseous exchange later. So how about alveolus? Okay, alveolus you should know about their because characteristic. They have a very large surface area. You are increasing the surface area to allow more diffusion of gas across the surface. Yeah, this is correct. And also you need to have a thin wall, right? Thin wall. Why? Because thin wall means shorter distance. Thin wall means shorter diffusion distance, which will increase the efficiency of transport. Yeah. You need to have a good penetration with air so they can maintain the diffusion gradients. And also you need to have a good blood supply. So that's why you can see the alveolus. Alveolus is being highly connected by a network of blood capillary. So those will provide a very good blood supply so they can maintain a very high convergence. So diffusion can occur faster. You can see this picture, right? This is how oxygenated blood is in. Here. Okay, actually these oxygenated bloods come out from where? From the right side of the heart. which is come from the pulmonary artery. So means that the oxygen blood from the pulmonary artery. And then when trouble pass by the alveolus, so they will release the carbon dioxide out. They just release the carbon dioxide out. Why? Because this place, they contain the higher pressure for the carbon dioxide. This place. They contain a higher concentration of carbon dioxide. I should say it this way, sorry. Higher concentration of carbon dioxide. So this higher concentration of carbon dioxide, they will just diffuse from the blood capillary to the alveolus. Yeah, they will just diffuse out. And also, alveolus now, they have a very high concentration for the oxygen. So now, alveolus, they have a very high concentration of oxygen, right? So now they need to release oxygen out because they can see that now red blood cells, they contain lesser gas inside. Yeah, so they will put the oxygen bursts out. Okay, then for this one, okay. Now we continue to talk about the structure of breathing system. Then for this one, you should know about the structure, okay? You should know how to identify the structure. The rib cage, intercostal muscle. Intercostal muscle is a muscle they found between the rib cage. This one, see? It's a muscle between the ribs and which control the movements, causing the inhalation and exhalation. So later when we try to do this inhalation and exhalation, what we do is this intercostal muscle, they help us to contract or relax. Okay, so ribs, the rib cage, what they do is, because rib is very hard, right? So rib, they can help to protect our internal organ, especially the hearts and the lungs. Yeah. And the diaphragm contains the sheet of the connective tissue and the muscle at the bottom of the thorax. They help to change the volume of thorax to allow inhalation and exhalation. It's referred to the connective tissue and the muscle. Yeah. So trachea is a windpipe. Trachea is a windpipe. And then voice box is larynx. And the bronchi is branching of the trachea with the one bronchus. Okay, I think that's all. And also remember that alveolus is a tiny air sac, just a 1 cell thick, right? The thickness. Very, very tiny only. Yeah, that's all. And now maybe we can talk about the intercostal muscle. Okay. Intercostal muscle, which is a muscle between our rib cage, we have two types, external and internal. So muscle only able to pull the bones but not push on them. So this means that there must be two sets of intercostal muscle, one to pull the ridge and another set to pull it down. So at the one thing you need to remember that whenever your muscle trying to produce a movement, right? So actually it's two pair of muscle working. It's not two pair, it's a pair. It means that two muscle working. Oh, maybe I can write this one. A pair of muscle, okay? What they do is they will contract and relax. A pair of muscle means that there's two muscle, right? Two muscle. One muscle contract, one muscle relax to produce movement. Means that when you're trying to contract, right? Maybe other muscle, they will do relaxations. They cannot contract in same time or relax in the same time. No. Because whenever they contract in same time, relax in the same time, they are unable to produce movement already. Okay, so you can see when they are flowing outside, it's called as external intercostal muscle. When they are flowing inside, is called as an internal intercostal muscle. See, this is the larynx, clavicle. Okay, and then the internal intercostal muscle. You can see this internal is lie really very internal. External lie outer bit. Yeah. And then this is for your information, okay? Trachea is windpipe, right? It's a C-shaped cartilage, yeah. C-shape. And they contain rings of rings of the cartilage, right lung and bronchus, bronchiole, and ribs. Okay, this one. Now we talk about the respiratory structure one by one. When we talk about the trachea, right? The trachea actually is being made from the rings of cartilage surround the trachea. Trachea is made from cartilage, right? Cartilage. What is cartilage? Cartilage is a kind of soft. It's not a bond. It's a very very soft structure, right? So you touch you touch your nose, right? This one. This is cartilage. This nose is made from the cartilage, like this. And then for this one, where is it? So cartilage is very we call it soft. Their character is they are very soft and light. And but cartilage is considered flexible, also flexible, and is slightly rigid. It means that the trachea is not soft at all, but the trachea itself is flexible. Okay, it's flexible and slightly rigid. So cartilage, which is a ring of cartilage, is they will surround the trachea and bronchi. Ring of cartilage, see, they surround the trachea and bronchi. So the function is they will help to support the airways. They want to support the airway and keep them open during breathing. So this is trachea. And if you're not present in the site, they may collapse inwards and the air pressures inside the tube drops. Okay, what does this mean? This means that, okay, maybe I can show trachea picture like this. Trachea picture. Trachea we call it windpipe. Why we call it windpipe? Windpipe is referred to the just like a water pipeline. See, water pipeline. Okay, so you can see that trachea is very unfortunately, trachea can be compressed too, like in this way. Okay, this is just for you to have a look on it. I can see this. This is trachea. I can see trachea. Trachea in the normal way, they should be look something like this. There's a long hollow tube, right? Passing from your mouth and turning into your thoracic cavity. So whenever the trachea they're being collapsed, right? We call it trachea collapse. I forget Mandarin call what. Just trachea collapse. Just trachea just being breakdown. Okay, it's not breakdown. It means that trachea being compressed. Compression of trachea, right? See, so you can see this dot. This is dot, right? So you can see the trachea become like compressed, almost no obvious hole. So this will cause because of breathing difficulty. Okay, see, this is trachea. So this is mean by what? Trachea collapse. This is mean by trachea collapse. So when you see the trachea, you see the lumen of trachea, they become smaller and smaller. You can see. In front still very wide, right? But below that it's become very narrow already. So you can see this is trachea. See, this is trachea. Interesting picture, right? I hope you feel that it's interesting. See, this is trachea. Same with us, okay? Our trachea is just look like this. This is requires a nasal cavity. Sorry, nasal cavity. We call it oral cavity. So actually, both cavity is connected to the trachea. This trachea, see? So this trachea, you can see, this is normal trachea, right? Why? Because that their lumen is still very wide, not narrow. So if they were not present in the sides, they may collapse inwards where the air pressure inside the tube drop. And then for tutor, okay, the function of cilia and mucus is, okay, along our respiratory tract, we have this cilia and mucus. This is both found in our respiratory tract. Include the trachea, bronchus, bronchiole. The passage down the lungs are lined with the stellate epithelial cell. See, there's cilia here. So cilia, they can do what? They can beating. They do the cilia beating, right? Cilia beating. Why they need to do cilia beating? Okay, Ethan, why they need to do cilia beating? What is the purpose that the cell need to do cilia beating? It's because that they want to do what? Cilia beating. They are trying to move the mucus. This is the purpose. Okay, they try to move the mucus. They try to sweep the mucus. Okay, so the mucus can cover a larger area. So after the mucus cover a larger area, right? So the mucus can trap. They can trap the pathogens. See. So cilia is they have a tiny hairs. They can beating means like they're moving. Okay, left, right, moving like this. So they can push the mucus up to the passage toward the nasal and throat, where it can be removed. So the mucus is made by the special mucus producing cell called the goblet cell because they shape like goblet or cup. Okay, the mucus trap particles, pathogen like bacteria and virus, and thus and prevent them from getting into the lungs and damaging the cell they are. So just need to remember about cilia and mucus functions. Okay, cilia is help to do the cilia beating, help to sweep the mucus. Like this, they beat and push the mucus up. See, they beat and they move the mucus up towards the nose and throat. Why? Because after that they can be removed by either coughing or sneezing, right? Mucus made from the goblet cell. Please remember this one is goblet cell. Because goblet cell means that the cell, they look like the cup. The cup. So they trap the particles, pathogen like bacteria and virus. Yeah. Okay, this one. Mucus is produced by goblet cell and trap the bacteria, dust, and particle. They allow to do the cilia beating and push your mucus away from the lung. Okay, that's all. So now we talk about inhalation, exhalation. We still have few more minutes. I think we still have time since we start late today. Inhalation, exhalation. What happened is because that you need to know about this one. Okay, maybe I show you this one. This. Where is it? I show about this. Okay, you may open your chapter 11 important past year note. Let me show you some. Easier for you to study. See this one. You can open this one. Important past year note. I also do send you in the WeChat group. Okay. Maybe you guys. Okay. You need to know about how do we do inhalation exhalations. Since this note didn't tell you in the clear way. This note. So you need to know about inhalation exhalation. What will happen? Okay, what will happen? See, inhalations and exhalations. What happens? So maybe we can start with inhalation first. During inhalations, okay, during inhalations, the air need to come into our lungs, right? So how the air enter into our lungs? That means that the air from the outside, they need to have a higher pressure. Air from the outside always come in with a higher pressure. So you just ensure that your thoracic cavity has a lower pressure compared to the outer pressure. You can force the air flow inside. Okay, so you can see that how you're able to reduce the pressure. First, you need to reduce pressure first. You need to reduce the pressure within the thoracic cavity first. What is because that when you reduce the pressure, you're actually increasing the volume. You're increasing the volume for the thoracic cavity. So how we're able to increase volume of thoracic cavity means that we need to we need to ask help from our organs. For example, our rib cage, right? Rib cage, they will move up and outwards. Okay. Our diaphragms also will contract and flatten. They will move down. Why they need to contract and flatten is because that they need to provide a space, okay, an area. Then pressure inside the thorax decrease. Air is flow in. Okay, so you can say you have these two muscle, external and internal. Okay, then you should know like why this muscle contract, why this muscle relax. So you can measure it this way. This is when I try to drop rib cage, right? This is in the intercostal muscle. Like this. Okay, so you just imagine that this orange color is referred to what? This orange color is referred to the external intercostal muscle. How about this blue color? This inner blue color segments is referred to the internal. So whenever you are trying to request pushing your rib cage to outwards position, means you push, right? You're pushing outwards as you need to increase the volume to reduce the pressure. So, yeah. So this will cause the contraction of the external intercostal muscle. See? Then, however, the internal muscle relax itself. So this will cause a pressure decrease and air come inside. Then how about acceleration? Okay, acceleration. The pressure will increase and the air is moved out onto the lungs. So the diaphragm will relax and move out. See, diaphragm will relax and move out. So you know that you know why they need to do the acceleration. I mean, no, you know how the acceleration happen. Acceleration happen is because that you need to remove the air away. How you remove the air away means that you need to have a higher pressure within your thoracic cavity compared to the pressure outside. Within a thoracic cavity. If you want to have higher pressure, that means that you need to reduce the volume. Means you need to reduce the volume first. Reduce volume for the thoracic cavity, make it become smaller, so you're able to increase the pressure. So whenever you do acceleration, right? Your thoracic cavity is actually pushing inwards. They are moving inwards, right? So whenever you want to move inward, right, your muscle also need to do the cooperation. So just similar with this, just this time, internal intercostal muscle are trying to contract and push inside. Yeah, okay. So it's like this. You need to know the term to write for the inhalation acceleration. Actually, this is like very easy question if they really come out in the exam. And then so you need to know how to explain now, okay, and how to describe. Maybe rib cage is like moving upwards and outwards, right? Okay, then for this one, difference between the inhale air and exhale air. What is the difference? What is different for the inhale and exhale air? Okay, you can imagine like this way. See, when I'm trying to do the inhalation, right, I can't feel nothing. I mean, I feel nothing, but actually, I already absorb a lot of oxygen, but I just feel nothing. But if you try to exhale your air, you see, you put your hands, okay, you put your hands in front of your mouth, right? You exhale, okay. What you feel? You are feeling actually a lot of water vapor. You're actually feeling water vapor from your hands. It's because that the exhale air, this one, the exhale air, they contain a lot of water vapor. See, This one. During exhalations, when you're trying to exhale the air, right, actually you have the higher percentage of the water vapors. Water vapor is a kind of gas, okay? It's kind of gas. So it means that the water, they will undergo the evaporations, then they become water vapors. It's because that the water, usually they'll evaporate from the moist lining of the alveolus. Remember that, okay? Remember that the alveolus, they have a very moist lining. Moist means what? Moist means the internal surface of alveolus is moist, means that they contain some moisture contents 就是说里面它这个表面有水分这样子 ，OK？ I mean not water just like 表面 is look like a little bit moisture. Okay, so that's why every time when you exhale the air out, the air they will pass by the alveolus, and alveolus is like moist lining, right? That's why when you exhale your air, they contain some percentage of water vapors. And then for the oxygen, right? Okay, oxygens, inspired air, they have more, 21%. Carbon dioxide, okay, aspired air, they have more, it's about 4%, yes. Then the inspired air, which is 2160, is because that oxygen is being removed from the blood by the respiratory cell, so the blood is returning to lung has lower oxygen concentrations than the air in the alveolus, which means the oxygen diffuse into the blood in the lung. Then how about carbon dioxide? Carbon dioxide is being produced by respiration. See, carbon dioxide is a byproduct for the respiration, and then they diffuse into the blood for the respiratory cell. So the blood transport the carbon dioxide to the lung where it's diffused into alveolus as in the higher concentration in the blood than the air in the alveolus. So just remember that when the alveolus contact with the blood capillary, right, usually the blood capillary contain a lot of carbon dioxide. Yeah, I think that's all. Only this nitrogen. Nitrogen gas is very it's very stable. Just 78 only. So very stable and cannot be used by the body. Yeah. Okay, that's all. You need to know about the reason. Okay, the reason happened behind the okay, then for this one, okay, use the lime water. This is a kind of experiment. They do it. Use the lime water to test for the carbon dioxide and the SUA. See, how do you do the lime water? Lime water is acidic or alkaline? Lime water, you think it's acidic or it's alkaline? Lime water is a bit okay. Maybe we have a look on this. See, why we use the lime water to test for the carbon dioxide in the SOA is because that we need to observe the difference between the amount of carbon dioxide in the inhaled SOA. So you have A and B. So when we breathe in, the air is drawn to the point tube A. When we breathe out, see, breathe in. Here is breathe in. Then here is breathe out. So when we breathe out, right, the air which is carbon dioxide, they will just go through the test tube B. So lime water is clear but become cloudy when the carbon dioxide is bubbled through it. This is lime water. So now after carbon dioxide enter, right, so they become cloudy already. The lime water in the body tube A remain clear, but B become cloudy. Why B become cloudy? Because it means that there's presence of carbon dioxide and it's a lot. It's a higher concentration of carbon dioxide compared to test tube A. That's why test tube A doesn't change color, just remain clear. Just in this one only. Okay, and then for this one, okay, investigate the effect of the exercise on the breathing. Okay, this one maybe we can have a look on this document. Where is it? The document that I sent to you just now. Oh, this one. Yeah, this one. So you should print in your mind. Inhalation and exhalation, what they do. Inhalation is referred to the external intercostal muscle, right? They contract. They contract so they can cause the thoracic cavity just pull outwards. So the wheelchair move outward and outwards, and then they contract, become flattened. So increase the volume. How about exhalation? Exhalation means that the external intercostal muscle, they relax, while the internal intercostal muscle, they contract. Exhalation, right? Exhalation means that your thoracic cavity is pulling inner, okay, pulling. So the wheelchair is able to move downward and inwards. So at the same time, the diaphragm must relax and curve outwards. Yeah. So those are to, because to reduce the, to increase the pressure, Increase the pressure, so you're able to decrease the volume. So it cause the pressure inside become high, so you can just breathe out from your lung. Okay, for this one they just show about the lactic acid. Okay, in the next chapter we'll talk about the lactic acid, how they being formed. So lactic acid is a product formed from the fermentation products. So this lactic acid, okay, they will produce when you are doing exercise, right? So that you feel your legs very, very pain due to the presence of lactic acid. But no worry, this lactic acid, they will be declined day by day. They will disappear later. Okay, so this is the study that we did. Okay, this is study that we did. See, initially when you start resting, right, after that, you do exercise already. So when you start to do exercise, you see the lactic acid, lactic acid, 乳酸, lactic acid, they just increase so fast until here already. When you recover, just drop. So this is graph we know as this one, this oxygen consumptions, oxygen deficit, this one. So this means what? This means that this show about, we call as the oxygen demand. Okay, oxygen demand. Yeah, so you can say that when you are starting doing the exercise, right? Okay, your oxygen become more deficit. Yeah. Okay, so this one you can have a read. Maybe I can discuss with you about the part that I look a bit hard for me. I think both of you also see. Just remember to read about the, we call as the notes. Okay. So this is the question they will ask in the exam. What is function of cartilage? So cartilage is a flexible, rigid. The other tissue. Keep the trachea open. Okay, this one. You see? Describe and explain how exercise affect the breathing rates. So when you are doing exercise, right, you are actually increasing the breathing rate. How you're able to increase? It's because that when you are doing exercise, you're increasing your respiration. And also you are increasing the energy demand for the muscle. Why? Because they need to do, they need to contract more. So you need to increase the respiration to increase the carbon dioxide in the blood. Okay, so the brain send me the nerve impulse to the external intercostal muscle and the diaphragm to contract to cause more regular and deeper breath. So you are increasing the rate and depth of the breathing. Means that when you are doing exercise, right? When you are doing exercise, as well as physical activity, you are actually increasing energy demand for the muscle cell. As the muscle need to work harder, right? They need to contract and relax more frequently. So you need to increase in respiration supply. Okay, as when the carbon dioxide level build up in the body, they will be detected by the medulla in the brain. So the brain send nerve impulse to the external intercostal muscle to contract, to cause more regular, deeper breaths. Yeah, means that this carbon dioxide, if they too much in the brain or in your body, so your body will be detects. Okay, detects. Usually how they detect is when they saw that the pH water decrease already, because that carbon dioxide is an acidic gas. Remember, carbon dioxide is a kind of acidic gas. So when they send the impulse to the external intercostal muscle, so they will contract. Yeah, they will contract because they need to pull inside. Okay, for this one, how heart rate increase when it's exercise, because that, and then you should know what kind of the method cause the increase in heart rate. Just traditionally, you need to exercise, right? But now you can use adrenaline. Okay, adrenaline is kind of drug, right? It's a kind of drug. Okay, they help to stimulate and increase the heart rates. So they can cause more muscle contraction as muscle require more energy, right? So you are actually increasing the cellular demand for the oxygens. Yeah, and you are also increasing the rate of the aerobic respirations because you are increasing more intake of the oxygen, right? So it cause more blood supply to the muscle, more blood containing the carbon dioxide supply to the lung. So this is the reason that why you need to increase the rate of the heart. 为什么你要增加那个心跳次数 ？If you increase the rate of heart, right, it will increase the frequency of pumping more oxygen blood to your whole body. So you hope, I mean, your body cell will not have suffering the lack of the oxygen. Okay, so this is the class for today. And then Zuyang, yeah, we finished. I think we haven't finished chapter 11. Actually, we still have a small part, okay, small part. Okay, I'll let you do the exercise first. I'll let you do the exercise first, chapter 11 until we still haven't finished this one, breathing. Yeah. Okay, so I will give you homework again. Okay, so there's a class for today. Thank you. Looking for your any question in future. Okay, bye. Bye bye.